We are moving on. Can I have the first slide of the clinical review and discussion on current paradigms of drug developments? Yes, Houston, we have a problem. Over the last 25 years, only 7 drugs have been approved. Only 7 drugs. And there is an estimate also reported by the Urlich-Germing-Düsseldorf group and others, that in about more than half of the patients, especially those with lower risk, after 2-3 years, we don't have any effective agent to offer. And this is in contrast with other myeloid or hematological neoplasms, as you can see here in the slide. For lymphoma, over this time, 17 drugs, myeloma, 15, AML, 12, CLL, 9, MDS, only 7. So we wondered what are the reasons, and I asked 2 top world experts to help me and to share their thoughts about it and to try to provide us explanations. Valeria Santini from Florence, who is here with me as a co-chair, will talk mainly about higher-risk patients, and she will explain why all these trials with high-risk patients failed. I will follow, focusing on lower risk and several other aspects of general development of drugs. And finally, Dr. David Steensma, who is well known as an MDS expert, but now as a chief medical officer in Ajax Therapeutics, will tell us the pharma's point of view. Valeria, please.
Thank you very much, Moshe. I will not explain. I will try to understand what happened and why. If I had the possibility to know why, maybe I would be a colleague of David or just having great results. So thank you, anyone. These are my disclosures. And also, all the drugs I will present in my lecture, in my talk, are not approved because of the failure. So these are not approved drug, and with the exception of azacitidine. Now, I drove this design some months ago, maybe more than a year ago, and I hoped that some of the drugs that are here in red would have become available and would have been successful in treating high-risk MDS. But as you see in the square, unfortunately, all the attempt of the combination of HMA with other agents were unsuccessful. And this is a very sad slide because I actually had it as my educational at ASH in 2012. And I am afraid that apart from some modification for the age of transplant-eligible patients, this is still unfortunately valid. Of course, you may say that we use IPSS-R and that we use IPSS-M, but the picture doesn't change much. So investigational agents, question mark. What about the standard of care we have for high risk, which is azacitidine or decitabine? You see these are real-world data that show you that when we treat patients in our centers who are not selected and who may have some comorbidity, the median survival in Italy and in the Netherlands is around in between 16 and 18 months. And this is not really a great achievement, but still shows you that we may prolong the expectant life. But then I show you a very depressing slide that is showing you that the overall survival in TP53 mutated patients, monoallelic versus biallelic, it's really bad. And the biallelic patients have a very short expectancy of survival, independent of all the therapy you can try. They may have a response, but the response is short, and therefore we do not have a real treatment. That's the worst we can have for high-risk MDS patients. And we may discuss also with AML. But then we had the hope to have a drug, APR-246, that was reactivating the function of TP53, because TP53 is mainly mutated as a loss of function. So there were 2 studies, head-to-head, phase 2, that showed indeed a very good response to this drug in patients with this mutation. And here comes the phase 3 trial. The phase 3 trial did not show overall response rate or overall survival difference among the patients treated with the azacitidine only or the combination with eprenetad POPT. So what next? So the combined results of these 2 studies have been published recently. And with a longer follow-up, we do not have any publication for the phase 3, in my knowledge. But what is the main observation here? Here we had more patients with biallelic TP53 than patients with monoallelic TP53, and this could have been the reason why these patients were so less responsive, and probably the drug was not enough to overcome the biallelic defect. So why these patients failed to show any response? Are these too high risk? Is transplant for this patient, with all the caveat, the only solution? Or should we look at this kind of therapy for TP53 mutated patients only as a bridge to transplant, because the responses don't last? These are actually open questions. And then we had the adventure with pevonedistat, is an inhibition of NET deletion combined with azacitidine. Phase 2 data, very, very promising. And then you go to the phase 3 data, showing, comparing the combination of pevonedistat and azacitidine to azacitidine alone. And the same was true for other studies. They combined higher risk MDS and CMML. And the stratification was there, but the population was sort of heterogeneous. And you see that the primary endpoint was event-free survival. What happened was that actually the response rate and the overall response rate, as you may see, was not really favoring the combination. And the event-free survival and overall survival, again, didn't show a significant difference. So no primary endpoint was met, although it was clear in a post-hoc analysis that if you prolong, the patients who had more cycles, so more than 6 cycles, may indeed show some advantage in overall survival from the combinations. So why is that? Lack of hematological response because the treatment was not long enough, or we selected the patients who anyhow had a lower risk in some sense. Do we need longer drug exposures? But then we anyway select the patients who somehow respond and survive. And then again, so another failure. We expected to have a completely new mechanism of action, anti-CD47 antibody. The magrolimab study was comparing the combination with azacitidine to the placebo plus azacitidine. And the idea was that this kind of reactivation of macrophage activity would overcome even the difference in mutations and the risk based on mutation. But unfortunately, when you look at the outcome, there was not an improvement in response. And unfortunately, the overall survival, as you may see, was really not favoring the combination, especially for the high mortality in the very first period of treatment. And as you see here, this was really probably the most important finding, that there was such a high toxicity in terms of on-target anemia that was in the beginning not really mitigated, that could have been impacting on the result. So the study was closed for futility. There was probably an under-evaluation of this toxicity. There was a bias in the enrollment of high-risk patients. I hope we will see the results published to understand more in detail what happened in this case as well. Okay, then we change gear again. Another mechanism of action, stimulus MDS2, was a study comparing the combination of sabatolimab plus azacitidine to placebo azacitidine. And sabatolimab is an anti-TIM3 antibody that reacts with exhausted T cells, but also with leukemic stem cells. And again, patients with CMML, high risk, and higher risk MDS. And the primary endpoint was overall survival. You see the responses were, in fact, favoring the combination somehow, but the overall survival primary endpoint, again, was not met, even if you consider MDS only. So the problem I'm showing you is that almost all the study in high-risk MDS combined high-risk MDS and high-risk CMML, which are not exactly the same kind of disease and may, in fact, lower the power, the strength of the study. But nevertheless, these are the data for the MDS only, so no difference in CR nor in overall survival. Is this a lack of proper selection? So the biomarker, the immunological pattern and characteristics were not considered enough to select patients. This is something we should always ask for all the studies I mentioned to you. What was the background biological basis for these treatments? But what we saw in the sabatolimab plus azacitidine post-hoc analysis was that women, in fact, had a statistical significantly longer overall survival. So sex in this case, and most probably the immunological asset and the T cell exhaustion was very important. And so a sex-based approach could also drive some new ideas and some new results. Finally, this is the result of an interesting study combining azacitidine with tamibarotene as a RAR-alpha inhibitor. And what was the purpose of this study? To see whether patients who overexpressed RAR-alpha, patients with high-risk MDS, could take advantage of the combination. And here, the primary endpoint is CR, is no longer overall survival. So the idea is that you sort of reactivate maturation the gene expression by combining tamibarotene with azacitidine. But as you see here, there was, in fact, an improvement in CR, but actually the study was not significant. So this study as well was failing, but we learned from this study and the other a lot, because we learned that we have really to organize our studies in a different way. And probably there was, at the basis of this failure, a lack of validation of the biomarker, the RAR-alpha overexpression in high-risk MDS. And what about the patients who had this overexpression? What was the exact role in high-risk biology? So the Verona, you will hear more about Dr. Garcia-Manero tomorrow, but the expectancy for this last study was very, very high. So we were anxiously waiting for the results, and the phase 3 Verona study combining venetoclax to azacitidine and comparing to azacitidine alone, unfortunately was not successful, meaning and stressing the point that AML and MDS are not the same disease and are not a continuum over. So the endpoint here was overall survival. You may note that in the majority of the studies I showed you, the TP53 percentage is pretty high. Here, a quarter of patients had TP53 mutation. And these were, of course, patients who had different percentage of blast and different IPSSR risk. No difference in overall survival when you take the entire population in intermediate and higher risk MDS patients. And there were no new safety signals, so no excessive toxicity, but again, you'll hear more about the Verona trial in this meeting. These were the results shown at the SOHO meeting last September. Higher marrow CR rates for the combination versus the azacitidine alone. But again, is this important in the overall result? Probably, as I said, biology is different. There was a lack of selection of the patients with probably higher risk and more blast in the marrow who were better responders. And again, are we using this combination as a bridge to transplant? Maybe this is the clue for these kind of very high-risk patients. So what are the lessons learned? Therapy should not be empirically based and just tried. We should have more insight into the biological background. And as a consequence of this insight, we should have more stringent selection of patients. There are no therapies that fit all patients and all molecular subtype of patients. Some agents are very active for a short while, so they can be considered mainly and only probably as a good bridge to transplant, and the duration of therapy is critical. And I really don't know what is the future now, if we will just get rid of the hypomethylating agents, find a new way. The triplets disappeared from our horizon. So we are actually desperately trying to understand and to learn and build on it. And I really wish to thank you for your attention.
Thank you, Valeria. I hoped you would give better answers, but we will try. I will move on, and I would like to share with you some thoughts about clinical trials in general, how we do it, and how we interpret it, and with some focus on low-risk MDS. Now, the way that we usually do, and this is how we were trained and how we are doing now for maybe 50 years, is basic research, whether it is from the laboratory, animal studies, a good idea, is followed by clinical trials, phase 1, 2, 3. And if and when they are successful, then there is a regulatory approval, FDA, EMA, and so on. And then the drug is marketed. And after all, the idea is to have the patients get the drug, not only the trials. I guess you would agree with me. So why sometimes things are not going well? Well, it could be that the drug is not effective enough, obviously. But there might be other reasons, probably related to the trials and maybe to what happens after that. And I would like here to focus on some problems, challenges, and limitations that we might come across during trials, and then talk about what happens after and when a trial is successful and the drug is approved. Finally, I would try to think about some possible recommendations. So let's evaluate the problems and limitations in clinical trials as we do it. One problem is the disease. The diseases, unfortunately, MDS is a very heterogeneous disease, variable biology, numerous genetic mutations, some of them we heard today, different treatment response patterns. Unfortunately, it is not like CML. I always look at my CML colleagues. Their life and their patient's life is very easy, very simple. One mutation, one treatment, and they are arguing between the 92 or 94% response rate. We wish we had been in the same situation. And there is still limited understanding of the biology. We have to take into consideration that the incidence is relatively low, which means that it is difficult to recruit patients for studies, longer enrollment periods. The result is a smaller sample size, which might lead to inconclusive results, and obviously the pharmas are less interested. But it's not only the disease. It's difficult to deal with these patients. They are relatively older. They are with comorbidity, as we discussed today. So that means that many of them are excluded from trials. There are logistic problems. They are less mobile. The tolerance is not always as good as we would like it to be, which leads to protocol violation. There is a higher dropout rate. And they are less available for long-term follow-up, which is more important in patients with lower risk disease. And obviously, there is the methodology of the trials. Many of the trials are too complex. The regulation is quite tough. We all know it. And let's think about the eligibility criteria, which are very, very heterogeneous. Are we dealing with lower transfusion burden or higher transfusion burden? Are we treating primary MDS, secondary MDS? These are all different. The endpoints are very important. What are the endpoints? Now, and the markers that we are using, you know, in the past, when there was cancer trials, survival was the only one marker. It was very simple, whether the patient was alive or dead. But today, look at this list. Complete response, partial response, hematologic improvement, PFS, event-free survival, leukemic transformation, MRD, and so on. And the trials are using different and various endpoints. Quality of life, how exactly there are some methods, but they are not the same, and it's quite difficult. One of the reasons why it is so complex, or one of the proof, you can see that how many modifications we are doing during the trial just to show that the design was not really appropriate from the beginning. Now let's talk for a minute about the statistics. Small sample size. The Nobel laureate Daniel Kahneman and Amos Tversky wrote a seminal paper, not on medicine, in 1971, and they called that the law of small numbers, which means we all know that if you take a small sample, then you are prone to bias with the results and the interpretation. And we often see subgroup analysis, not always they are right. Post-hoc analysis, I will show you also, but we know the limitations. And let's say one word about the holy p-value. We were trained by the binary way. Either the p is less than 0.05, successful, wonderful. And if the p is 1.51, 52, bad, failure. Well, life is more complicated. It's not like this. One excellent editorial in New England Journal of Medicine, Evidence, about a year ago, the title was Treat Patients, Not P-Values. And another report shows that if the p-value, even if it's successful, and it is close to 0.05, if you want to repeat the result, you need 16-fold larger sample for that. So we have to be careful when we look at the p-value, when obviously it might lead to misinterpretation. Now, with the interest of time, I won't show you many examples, but I will just focus on very few, not in the MDS fields. Many of the COVID trials have been criticized, especially by the NIH. There is here an alliance study with breast cancer, which was defined as failure, but the editorial shows that some subgroups did benefit from the treatment. I was involved in some of the erythropoietin trials, and it was sort of risky drug, and the oncology solid tumor do not use it anymore. I'm not sure that this is the right, because there are proof that part of the problem with erythropoietin was allowing hemoglobin to go too high, not that it stimulated cancer. and there are several other examples. But there are also examples in the MDS field. Valeria mentioned the pevonedistat, first the Panther trial, but interesting point which I would like to emphasize, I think Mikhail Sekeres presented in one of the ASH that in some subgroup, the VAF went down. So probably if the VAF went down, it might indicate that at least some subgroups might benefit from a trial that was defined as a failure and is not developed anymore. We are familiar with the thrombopoietin receptor agonists. They are not developed anymore, but there are probably 2 trials, and it is clear that at least in lower risk, it might be safe. I was privileged to lead the roxadustat trial, which was also defined as a failure. But just to tell you, I have several patients on this drug, and at least one patient went from hemoglobin of 7.5 to hemoglobin of 14. I am not aware of any placebo that might increase hemoglobin. So maybe the drug is not effective for all, maybe only for subgroups, maybe not the duration is not long enough, but obviously there are some patients that might respond. Initially, the company wanted to close the program. Now probably there is a future for this drug for new treatments. And there are several other examples saying that probably we should be careful in defining failure or success. Now there are other aspects which I will just touch briefly, such as the safety. Safety are not always reported as well. In one trial, summarizing 4 of 7 trials with more than 300,000 patients, it was clear that only 32% reported completely the toxicity. Emma Burat and colleagues reported recently that only 13% correspond with the inclusion of the toxicity. So toxicity is not always reported as it should have been. Now let's talk for a couple of minutes about what happened if and when a drug is defined as successful and is approved by the regulatory agents. Now, there are many gaps. There is a gap between the trials and real world, as we all know. One report, for example, showed that only 32% of cancer drugs that were approved after accelerated phase, indeed prolonged survival. There is a gap which is basically because real world is not what we see in trials. It is estimated that in the US, only 7% of cancer patients enroll in trials, and I can conclude that in many other countries in the world, it might be even a smaller percentage. There are certain underrepresented populations or subpopulations, minorities, people with low income, people in the periphery, in the community because they are far from the hospital, and in other countries. So they are not represented in trials, and we tend to conclude what happens in the trial is real world, which is not the case. We are all aware that industry trials are different from academic trials. They tend to recruit younger patients, healthier patients. The follow-up is shorter, and the outcomes tend to be superior, but this is not always real life. And also, novel information that is published later after the trial is completed might change things. There is limited commercial interest for some agents, especially in our field. And we also have to remember the financial toxicity. What happens to a drug that is approved, but if it is too expensive, either it is not covered or the patients cannot afford it. So what have we achieved? So, what can we do in order to change and improve it? Well, I am for evolution and not revolution, and probably we can do some things together to improve it. It should be a joint effort of all stakeholders, the academy, the pharma, regulatory, the society, and the patients. We got to better understand the biology and to move on and identifying better targets and markers. We have to improve and make the trials more simple protocols, broaden the eligibility criteria to include almost all, and by that, hopefully, the trials will be closer to real world. We have to also address the post-approval barriers, there are several programs for orphan drug. Some of them are outdated. They should be modernized according to the new tools. AI might help us in designing better trials and solving these problems. And also, we have to address the issue of cost as well as the toxicity, which I mentioned earlier. So, in summary, we still have a problem, but if we all work together and try to solve and to attack these limitations, we might come up in the next few years with better solutions for our patient, and the number of 7 might increase. And with that, I'll finish my part. This is what happened to one of my patients who had a prescription. The price may make you dizzy. And thank you for listening to me. And thank you. I would invite Dr. David Steensma, who will tell us about the pharma's perspective. David.
All right. Thank you, Moshe. So I should make it clear right at the beginning here, I'm speaking on behalf of my own idiosyncratic perspective and not in any sort of official role, giving some sort of unified position for how industry is thinking about drug development and MDS and what's gone wrong over the last 15 years with this drumbeat of negative trials. And I think Valeria and Moshe summarized it very well. It's a little like the opening lines of Anna Karenina, where Tolstoy wrote, happy families are all alike, unhappy families are unhappy each in their own way. And each of these different trials, it was a different problem. It was a study design, it was a drug that wasn't active, it was the wrong patient population. COVID got in the way of a couple of the trials. So that all adds up, but the dysfunction is not just a single thing that we can fix. But certainly it is very frustrating. In fact, there was a lot of hope for the Verona trial for venetoclax that, because that's made such a difference in AML, maybe it would similarly have made a difference in MDS. And we can talk about why that study was negative, but certainly it cast a pall on industry's perspective of developing an MDS. In fact, shortly after that trial completed, I was talking to a pharmaceutical executive at a big company, and I said, what are you guys thinking now about doing trials in MDS? And he said, well, we'd rather put a large stack of money on a rusty old barge and send it out into Boston Harbor, because at least when the barge sinks, some of the $100 bills would wash up on the shore and we wouldn't lose absolutely everything as we have for many of these trials. And yeah, that's tongue-in-cheek, but I think it does express a level of frustration. Nobody's running out and saying, hey, I want to do the next big trial in MDS and spend $50 million and have nothing to show for it at the end of the day. Some of these drugs, as Valeria pointed out, might be salvaged. Potentially, what if you did the sabatolimab trial again, but only did it in women? Maybe that would be a positive study. But a lot of these companies just turn the page and lose interest. And so that's the reality that we have to deal with. So yeah, 17 trials, to my count. The only higher risk trial in MDS that's been positive, randomized in the last 20 years was the transplant trial from CIBMTR and the CTN Transplant Network. So we should transplant people if we can. It's hard to look at these kinds of figures and look at like everything that's happened in lymphoma and myeloma. Comparison is the thief of joy. Is there something inherently more difficult about MDS that's made it harder to develop? Are we, as an MDS community, more dysfunctional in some way than these other communities? I don't think so. I think there's been a lot of bad luck here. And as my colleagues pointed out, there are some things about MDS that are particularly tough, such as lack of good targets and an older population. Hopefully these things will improve. You know, when industry looks at MDS, it has been looked at as a big opportunity. You look at the market size for the lymphoproliferative disorders for myeloma, and these drug approvals have resulted in huge global sales, but MDS is a pittance compared to that, even with a couple approvals in lower risk MDS in the last few years. There are a lot of different concerns now that pharmaceutical sponsors have about MDS, and many of these my colleagues have gone through, so I won't go through them in a lot of detail, but I'll just point out that it is really hard, you know, when preclinical models don't predict things very well, when a lot of the mutations that have been successfully targeted in other diseases aren't present in this disease or are rare in this disease, and we really don't understand the biology as well maybe as some other diseases. But I think we're a lot better than we were 10 years ago about biological understanding and about modeling. And, you know, a lot of that comes down to funding. You know, like the Evans Foundation has built a huge consortium of investigators who've made some real discoveries in MDS, and so hopefully that will continue. A big issue is that a lot of small trials looked good, but then the big trials crashed and burned, and you know, we can talk about why that is. You know, some of the things that aren't as much of a concern for industry, the heterogeneity, and as Moshe pointed out, MDS is very heterogeneous, but that's true of lots of disorders, and we can often still find a red thread tying the disease together that's targetable. There is a lot of MDS here in the US and globally, and so it's not that it's an ultra-rare niche indication where there wouldn't be a good return on investment. And it's not that existing therapies are so hard to beat. One of the problems that's happening in myeloma is that, you know, you've got BCMA-directed CAR T therapies and all of these different combinations. These are really, really effective therapies, and it's getting harder to show a benefit, so you have to look at surrogate markers like MRD and you know, progression rather than survival. That was the problem in large cell lymphoma for a long time. It's just really hard to get anything better than R-CHOP. But azacitidine is not that great. I mean, you know, 15-20% CR rate and median survival a year or two. So that hasn't been an issue as well. We have certainly seen this regression in the mean. I liked your reference, Moshe, to the law of small numbers. It is hard sometimes when you do a very early trial to know when is the response rate good enough to say, hey, this is worth doing a registration trial because it has a good chance of being successful. We've seen significant regression to the mean with several of these agents where, you know, there were phase 1b or phase 2 trials that showed high response rate and then that regressed. Not so much with venetoclax. The response rate, at least from the press release, we'll hear more at the meeting, was quite similar to what had initially been presented. But their cytopenias and infections, I suspect, were a problem. Andy Brunner at MGH and I and a statistical colleague did an analysis of, you know, if you do have a small study that has 15, 20, 25 patients, when is this response rate good enough to suggest it's worth taking on a big study? And so this could be a guideline for thinking about that. I think Valeria touched on the fact that AZA-001, which led to the AZA approval, really was an outlier and a bit of an anomaly. A lot of real-world data hasn't shown the same sort of effectiveness. I think there have been, you know, various mistakes. I mentioned jumping into big trials too quickly. A big problem is not listening to the MDS community. There have been companies who, you know, just decided they were going to do things and didn't talk to disease experts. I remember getting a trial to review from a company that had only done solid tumors, and, you know, they were proposing doing this trial with an immune checkpoint inhibitor that had been successful in lung cancer. And their MDS trial excluded patients with cytopenias. You know, they had no idea what MDS even was. You know, the endpoints were all wonky. And, you know, a 30-minute discussion with somebody like Raf or Ariane could have set them straight. So there is a little bit of that, maybe. There's also fear of missing out. You know, sometimes whether it's an immune checkpoint inhibitor or an HDAC inhibitor. One company does something, it looks exciting, and then everybody else wants to jump in the pool. That's not true just of MDS, certainly. How many different BCMA-directed therapies, how many different calreticulin-directed therapies are there now in development in MPN? There's just a lot of fear of missing out on something big. Poor trial design, like this elevator. Some trials have been very good and yet still have been unsuccessful. One of the things I think has been a recurrent trial design issue has been this lack of clarity how to do dose reductions. And that was a problem with Verona. Do you dose reduce the venetoclax? Do you dose reduce the azacitidine? Which one do you hold? I think that's a real problem. I just want to call attention to this paper. We don't have time to go through it in detail, but it does have some, I think, useful practical suggestions for drug development in MDS. So in the interest of time, I'll just wrap things up. But, I don't think it's hopeless, and there are still a lot of trials going on. Hopefully, we'll see some successful approvals in the next 5 or 6 years. But it just really has been, I think for all of us, very frustrating that we're not in a better position for our patients than we are currently. Thank you.